Welcome back, biology students. I'm Mr. Silverstein. Today we're going to get into part 3 of our discussion of enzymes and biochemical reactions. So get out your notepad, get out your spiral notebook, a couple of pens. I'm going to share my screen and let's get started. So we go back to our essential question: how do living organisms obtain, store, and use energy? Now, we haven't gotten to the energy part yet. We're still working on how the enzymes help those reactions get started. But our objective today, and in this lesson, is for you to be able to explain how an enzyme can denature or deform, and how different conditions affect the rate of reactions and enzymes help. This is the third part of this topic. There'll be a practice assignment at the end, and then we'll talk about our lab prep for our next live session. Let's get started. And remember, these slides are posted for you on your Google Classroom. Make sure you go back to them, rewatch the videos, and update your notes. So let's review part 2. Metabolic reactions are controlled by enzymes. Enzymes are mostly proteins that speed up biochemical reactions by lowering the activation energy needed to make those reactions happen. Because they speed up the reactions, they're called catalysts, and catalysts are substances that speed up the reactions without being permanently altered. Enzymes are very specific, and they have an active site that fits only one substrate, and the substrate is also known as a reactant. And we call that a lock and key model. And enzymes can be used to make or break chemical bonds. Let's go back and take a look at these enzymes. Enzymes are very specific. They have an active site that fits only one substrate. And once the substrate connects, the bind tightens up into what's known as an induced fit. And we use this illustration for the lock and key model of an enzyme. You have your enzyme, your active site, and your substrate or your reactant. But sometimes an enzyme can denature or denaturation. Basically changes its shape. And the enzyme's active site gets deformed and loses its specific shape. And when it loses that shape, you have a loss of biological activity. And this is caused by environmental changes like extreme changes in pH, a temperature increasing or decreasing, ion strength, and solubility. Now, what happens here is when the shape changes, the enzyme can no longer function well and the reaction slows down. So if we go back and look at our illustration on the left here, we have our substrate and our active site, and it's very specific. And if it denatures, it now changes shape. Now, some of the substrate will fit, but some of it will not. And if it can't get the full enzyme shape, it won't be able to create that reaction as quickly, or it loses biological activity. So here's our normal enzyme, and here's our denatured enzyme. And we're going to use these illustrations on your assignments and your exams, so pay attention to them. Some enzymes can be renatured back to their original shape, but not always. And if the denaturing agent was removed, the original attractions between the amino acids will reshape the protein, making it able to perform its natural function when it was still biologically active. But sometimes it cannot be renatured. Here's your normal enzyme. Here's your denatured enzyme. So the enzyme can denature, and if whatever was interfering with the original enzyme is removed, it may go back to looking like a normal enzyme again. Let's take a look at this video on how enzymes denature, and we're going to watch this together.
Denaturation of enzymes. Before we start, you should already know what enzymes actually are. Watch our video, What are enzymes? Each enzyme has conditions under which it functions best. In other words, the point at which the rate of reaction is at its highest. Amongst these conditions, the most well-known are temperature and pH. If these conditions vary from the optimal level for the enzyme can become denatured, and consequently the rate of reaction will decrease. However, different enzymes have different optimal conditions. One consequence of denaturation can be disturbance of the organism's inner balance, also called homeostasis. For example, the activity of proteins, which is not optimal, can affect protein digestion in the body. If we look in more detail, denaturation of enzymes and any other protein means that the shape of the enzyme has changed in some way, usually a permanent change. The activity of an enzyme depends heavily on its shape, particularly of the active site which binds the substrate or substrates. This change of structure means that the active site is not shaped for optimal rate of reaction. So, for example, if the temperature of the environment is too high for the specific enzyme, it may become denatured. Typically, enzymes that operate in warm-blooded animals tend to have an optimal temperature of 37 degrees Celsius, which is body temperature. Then as the temperature rises above that, the rate of reaction rapidly falls because the active site has changed shape. Whereas enzymes in bacteria that live in the thermal vents have a much higher optimal temperature. Now, here comes the chemistry behind this. When temperature increases, the individual amino acids that make up the enzyme vibrate at a higher frequency. This results in breaking of hydrogen bonds between amino acids and different parts of the protein chain, leading to a change of shape. At even higher temperatures, bonds between individual amino acids called peptide acids can break, leading the protein to fragment. Another important variable is pH. A pH level which is either too high or too low for a particular enzyme can lead to change of protein structure. Different enzymes will have different optimal pHs. The optimal pH for an enzyme depends upon where it normally works. Pepsin works in the acidic stomach, and so unsurprisingly has an optimal pH of 2. How does pH affect the active site of enzymes? If the pH is too low for the enzyme, then the concentration of hydrogen ions will be greater than normal, and these will interact with amino acids, leading to a change in shape of the active site, thus reducing the rate of reaction. So from this video, you should know that enzymes have optimal conditions under which they operate. Different enzymes have different preferred conditions, with some preferring acidic conditions and others preferring alkali conditions. Temperature is also an important factor, with most enzymes having an optimal temperature of 37 degrees and then denaturing above that.
So enzymes increase the rate of reactions, but other things can change the rate as well. When the enzyme denatures, Some of the biochemical reactions continue, but mostly they'll slow down. But there are some other things that can affect the rate of the reaction. Let's take a look. Some of the ways that change the rate of the chemical reaction include temperature. Increasing the temperature increases the rate of the reaction. Molecules are moving faster and colliding more with each other, and that makes that reaction occur much quicker. If the pH is changed, that will affect the chemical reaction of how acidic a solution is. Most enzymes only work at a very specific pH, so if the pH changes, it can affect the speed of that reaction. It can either increase or decrease. A third way is the substrate concentration. The higher the amount of substrate, the faster the reaction. The lower the amount of substrate, the slower the reaction. This is due to more particle collisions. So the more substrate you can add, the more particle collisions are going to happen, the faster that the reaction will occur. Then, of course, we add catalysts like enzymes. They speed up the reactions. They lower activation energy needed for the reaction to start. Now, we haven't talked about this one yet, but it is known as the competitive inhibitor. This is where something comes in and competes with that active site on the enzyme, and it slows down the reaction. When it competes with the substrate for the active site on the enzyme, it will slow down that reaction. It's kind of like someone bumping into you while you're working on something, and it slows you down. Or someone who comes into a conversation and slows down that conversation. When we do our lab activity, we'll be working with competitive inhibitors. Let's take a look at a competitive inhibitor and an enzyme function. This should take about a minute. Let's watch this video together.
During the normal enzyme catalytic cycle, the substrate encounters an enzyme with a specific active site to which it binds, forming an enzyme-substrate complex. The enzyme then facilitates the breakdown of the substrate to its products, which part from the enzyme, leaving the active site free to catalyze another substrate as the cycle begins again. Competitive inhibition occurs when an enzyme encounters a blocker, which mimics the properties of the substrate and binds to the enzyme's active site. Thus, when the substrate is encountered, the active site is not available for attachment and no reaction will occur. Non-competitive enzyme inhibition involves the binding of a blocker to the enzyme away from the active site. This binding causes a conformational change in the enzyme, altering the shape of the active site, which prevents the substrate from binding. No reaction will occur as long as a non-competitive blocker is bound to the enzyme.
So let's review. Enzymes can lose their shape and thus lose their effectiveness. And denaturation can result from high heat or pH change. Enzymes can also have competitive inhibitors that interfere with the enzyme's actions. And this can also happen from high heat or high pH. And the rate of chemical reactions change based upon temperature, pH, amount of solute or substrate, catalysts, and competitive inhibitors. All right. We've worked on this topic for 3 parts. What I'd like you to do next is to open up your assignment tab and open up the practice putting it all together enzymes. This is all going to be written responses. Please make sure you use full sentences, punctuation, and spelling does count. Next week in class, we will do a lab activity known as simulating a chemical reaction. And what I'd like you to do before you get to class is to review the reaction on page 6 through page 9 in your packet that you received this week. I'll also post that lab on Google Classroom for you so you can get a look at it. Well, I'm Mr. Silverstein, and that's all I have for you now. If you have any questions along the way, please email me and we'll get back to you promptly. If there's nothing else, you guys go off and have a great day, and we'll see you next time.